now for our last presentation, you guys get a 2-for-1. So both Elliot Fair and Carlin will be presenting to you because they work very closely on this one project together, and they are from my lab.
Hello, everybody. My name is Elliot Fair. This is Carlin, and our project was assessing the genetic relationships of wild rice populations within Michigan. So first off, wild rice is a very culturally important food to the Anishinaabe people of the Great Lakes region. It's known as monoman or the good berry, and it was a staple to their diet for hundreds of years. But within recent decades, the population is in decline due to poor water quality, pollution, and habitat loss. So right here is a wild rice species range map. So what it shows is that there's multiple species in the United States. The yellow, which is the Aquatica, and then the red is the Polustrus, and then the green area is a mixture of both Aquatica and Polustrus. So the two species we're working with in Michigan is Aquatica and Polustrus. They're physically different. Like Aquatica is much larger. I mean, it can grow almost as tall as me. Polustrus is not as tall, like only a few feet or a couple feet. But they're both found in similar habitat. Like, I mean, we found it anywhere from a couple inches to a few feet of water. So like I said before, both species are physically different, but genetically there's no clear separation among them. So like when we look at this haploid type network, it shows all the chloroplasts, like this is all from chloroplast DNA. So what we would expect if both are their own distinct species is something like the latifolia, where it's just a clear separation. But instead, we get a mixture between the two. So like the Polustrus is the circles with the hashes, and then the white circles is the Aquatica. So you just get a kind of mingling of the two. And so that either can be like the start of speciation, where they're starting to branch off into their own groups, or it could be a sign of hybridization.
So now that Elliot's given you a little bit of background on wild rice, we're going to tell you the focus of our project and what we did with these different species of wild rice. So the focus of our project was to see if these are genetically distinct species. As Elliot has told you, there is distinct physical traits of this wild rice, but the genetic story gets a little bit jumbled, as he showed you with that map. Also, to add on to that, Alex Wheaton from the environmental department at the environmental portion of the Gun Lake Tribe has said that in his work with wild rice, like he's having trouble even differentiating between the two species. Even like just looking at it, he won't be able to know which one's Polustrus, which one's Aquatica, etc. So even that portion's starting to get a little bit harder to distinguish. Also, we want to see, because of these physical differences and these genetic differences, can we line this up and see what's going on with that lineage there? Finally, if these are different species, is there hybridization occurring? And in order to find out the answers to these questions, we are performing 2 different types of sequencing. The first type of sequencing is the chloroplast sequencing, which gives you just the information from the maternal lineage side of the wild rice species. And this has been done in the past on other populations of wild rice that we compare it to. And the type of sequencing that has never been done on wild rice is the nuclear sequencing, which looks at both the parentage of the lineage of wild rice. And hopefully by understanding and analyzing this data, we can get a better idea of the wild rice in Michigan, if there's 2 species, its hybridization, and how this can help us better manage the species. So to break down kind of the steps of this project, our first step was to go to 14 different sites in Michigan to look at those populations. At each site, we collected 20 samples to give a total of 280 samples of wild rice. From there, we sequenced 3 different genes of the chloroplast using the chloroplast sequencing. And again, that shows the maternal lineage of that of the species. After we looked at that type of sequencing, we used rad sequencing, which is again the type that has never been used on wild rice before. So that took a little bit of trial and error. And we do that to look at a small portion of the genome, which again looks at the maternal and paternal side of the lineage. And once we have that data from the lab, we can then combine genotyping with phenotyping for each population to confirm that genetic lineage.
So right here are all the sites in Michigan that we went to. The reason why we want to show this to you is because it can just show like the broad range, like the area that we covered throughout Michigan. Now when you see like the lakes, we didn't give them their actual names only to protect those populations of rice there. And like the reason why we give you the river's name is we're confident that because of like the geographic area they cover, that no one's going to find the rice without special knowledge. And in these photos is just when we were sampling at a lake, and then down in there, that corner, that's the whole plant itself. And the reason why we at that specific lake we had to take out the whole plant because they treated it for like aquatic vegetation because they don't want it in that lake for some reason. And so what the Gun Lake tribe wanted us to do was collect it. And then they took it off-site, and then they're currently growing it themselves, and it's doing pretty well. And then right here is just a video of what we were doing, like how we were sampling. So you can see a pink flag down there. That's to help us with collecting phenotype data, which is next week. So it really is just there to so we can find the same plant potentially that we took the tissue samples from. And all we did for the tissue samples was we just had a pair of scissors, and then we cut off a little snippet, and then we just put it in a bag and labeled it.
So I mentioned the two different types of sequencing approaches that we're taking, and I'm going to get a little bit more detailed with that. So the first one is with the chloroplast sequencing, and the very first step with that is to extract the DNA. So what we did with that is we took the sample that we got from out in the field, we pounded it down to get past the cell wall, and then from there we did the chemical process of extracting that DNA. What shows here is the gel analysis of that extraction. And from here, we're going to look at two different things. So the first thing is to make sure that we actually have that extracted DNA present. And the other is to make sure that we don't have any degraded DNA present. And the reason for that is because it gives us a lower quality of sequencing data later on. So we know that there is degraded DNA present if there is a faded band line, like you can see right here, and then smearing down the band. So that's what we don't want, and we want to avoid using that later on in the steps. The second step is to PCR 3 chloroplast genes. So PCR, as some groups have mentioned earlier, is a method that we use to copy the DNA. So we make a bunch of those copies so that we're able to sequence those 3 genes later on. It also uses a certain number of cycles that creates more and more DNA with each cycle. The third step is to actually sequence the DNA. So the picture right here is just like a little snippet from the sequencer that we have with some of the nucleotides you can see there. And the final step is taking that sequencing data that we use using 2 different types of programs that will align the sequencing data and find the similarities between them and group them. So it'll find the ones that have some of the mutations, like the polymorphisms and things like that, and group them based off of that. And again, that has a checkmark /X because we have to get that phenotypic data in order to confirm this. And the second approach that has not been done on wild rice for rads is rad sequencing. So the very first step of that is the same as doing the chloroplast sequencing. So we extract the DNA, make sure there's no degradation present. The second step is to use a restriction enzyme digest. So this one specifically used the ALF1 enzyme, and it makes 70,000 cuts within the genome. And this gel here we use in order to make sure that the digestion did work. So you can see a more solid, you can see a more solid line at the top, which is just the regular DNA that we use as a control, and then the more faded one that's smeared down, which means that we did get digestion. So this one that I put a little bit too early is the ligation amplification step that we do next. So what we do here is we use a 1/4 reduction gene to find 2,000 regions that will be different between the samples. So it has the number of cycles here because we're still troubleshooting this part of the sequencing. We haven't quite finished yet, which again is why this has the check mark/X by it. So what we want to see here in the gel right here is to get the optimal number of cycles that I mentioned with PCR earlier. So right here, you can see that there is nothing present right here. We don't want that, but at the same time, we don't want all of the different bands that you see at n equals 25. So we know that the sweet spot here is more so in this area. Once we have perfected that portion of the protocol, we can actually send it off for Illumina sequencing. So what we plan to do is take a certain number of our samples, just a small amount, and sequence it here in the lab. And because we have 280 samples, we can't do them all here, so we will send the rest off to be sequenced. So this is the phylogenetic tree that we were able to produce from our sequencing data. So if there were two distinct species of wild rice, this is what we would expect to get between this tree, how you see these two different groups. So this first top group here corresponded to the southern region of Michigan that we looked at. The second group at the bottom corresponded with the more northern region. You can notice right here, these two didn't really box up with any. That's because one population matched up with the southern region, while the other one matched up with the northern. It should also be noted with this other region down here, is that some of the populations did correspond with those southern regions as well. So there was a little bit of mixing in there.
So right here is a chloroplast genetic variant map. So really what we're just looking at is all the genetic variances from the wild rice that we got from our sequencing data. And then each circle on this map represents a different genetic variant. And then each, like the size of the circle just shows you how many population, like samples from each population are in that genetic variant. And as you can see, some like circles have multiple sample sites in it. And then some sample sites have like multiple genetic variances, like Kalamazoo, which is this purple, which there's 4 of them. So that's just really neat to see how, like, in a small area, it has 4 different genetic variances of wild rice.
So as you can probably conclude from our other slides, our work is not finished yet. So some of the things that we're going to be doing in the upcoming weeks to finish up our project is to continue the field work to get that phenotypic data. We're going to observe some of the plant, how tall it is, the blade width, and then some other traits that are specific to wild rice that we're going to look at and compare between the populations. And then we'll compare that with the lab work that we conducted this summer. After that, we're going to perfect the red sequencing protocol for that ligation and amplification step, and then send off those samples that we took for red sequencing. Once we have all of that information finally, we can kind of line up all of our data and make those connections, and hopefully that will help with future management protocols with the Aquatica being endangered.
So really, what are the implications of all the data that we're getting from this study? So really, it's meant to help with conservation and restoration of wild rice. So like right now, we're trying to identify if there's 2 species of rice, which is what like we currently believe. And but maybe there's only one. Maybe there's a hybridization occurring, because let's say if we do find that both Aquatica and Plustrus are their own separate species, most likely the management plans for those species wouldn't change. But if it's just one species, then we might not even have to worry about it at all. But and if there's like hybridization occurring, we just have to determine whether that's good or bad. But previous studies have shown that most likely it's not a good thing because it produces less like seeds. So it's harder for it to like just grow. And so yeah, that's about it.
Okay, and we'd like to give our thanks to a couple different people before we finish up here. So our mentor and PI, Dr. Charlyn Partridge, oversaw the project, and she's been an amazing mentor and has done an incredible job this summer. We'd also like to thank the Jackson Lab, including Sindel, Maggie, and Renee. You guys all made like a great environment this summer and made everything a ton of fun. I really appreciate all that. We'd also like to thank Alex Wheaton from the Gun Lake Environmental Department and AWRI, as well as Pierce Cedar Creek for funding our internship this summer. For Carlin and Elliot.
May go beyond the scope of what you guys did. It's great, by the way. But in terms of the cultural significance of wild rice, which is obviously very large, how are you working with the tribe to figure out the management and significance of whatever your results might be?
Well, I guess what we would be doing is, so like, obviously we would be, we're working like significantly, I can't think of the word I'm thinking of. We're working a lot with the Gun Lake tribe on this, and especially Alex, because like that's his main job at the tribe is to work on wild rice. So it's just kind of like keeping that partnership going so that we can like just trade information, and so it can help just future efforts.
Yeah, and like whenever we did field work or we were like working on something new, he always attended with us with the field work. We're in constant communications with how things are going. So far, we haven't been able to give him much information, obviously, because we're not finished with our work and we can't really say what the difference is between wild rice, but hopefully we'll be able to change that soon.
But also, like I should mention, I am part of the Gun Lake tribe. So like kind of just like a personal experience, like everyone in the tribe really wants to see this crop come back and like explode so that we can harvest it all together. Like it's just a very big deal within like the personal like circles in the tribe.
I appreciate that. I don't know if you're aware, but we tried to introduce it in Stevie Lake about 15 years ago, and the avian population just totally embraced it down. So we didn't have any luck at all.
Yeah, I mean, ducks love it. What can I say?
Any other questions?
This would be a really silly difference, but were there like chromosome number differences among samples?
We didn't look at that.
Yeah, I'm not sure.
I was just curious.
Yeah, that's a little separate, but I appreciate the question.
Yeah. All right, so take a moment to thank all of our presenters today. All for coming
And thanks, Roxana.
and thanks Roxana for setting up the food and snacks and organizing it and sending out all the emails for everything, reminding everyone everything. So thank you all. We appreciate it.
Thanks,
Thank
Charlyn.
you.